I have no conflict of interest to disclose, and thank you organizers for this opportunity to share my work with you. It has been 40 years since first aid cases were reported, but still HIV cure remains elusive. One of the greatest challenges to date is the ability of HIV to establish a latent reservoir. In this transcriptionally silent state, HIV cannot be cleared by the immune system or antiretroviral therapy, necessitating lifelong antiretroviral treatment. One of the most promising strategies to date to target the HIV reservoir is termed as shock and kill. In this strategy, latency reversing agents such as panobinostat, prostatin, and TNF-alpha are used to shock cells containing latent proviruses so that they can be killed by the immune system or die from viral cytopathic effects. However, these efforts have been unsuccessful so far in impacting the sizes of latent reservoirs, calling for further understanding of viral and/or host factors in establishment and maintenance of latency. With regard to viral factors, HIV encodes Nef accessory protein, which is endowed with plethora of functions, making it crucial for viral replication and pathogenesis. Given that Nef is expressed early during viral life cycle and has an ability to modulate T-cell signaling events, we were intrigued to understand the role of Nef during viral latency and reactivation. To do this, we generated latent infected T-cell clone harboring a provirus with functional Nef, and also generated a corresponding clone without Nef through CRISPR-Cas9 gene editing strategy. And after that, we treated this cell with latency reversing agents. 24 hours later, we analyzed for viral reactivation by looking at GAG expression through flow cytometry. Interestingly, we observed higher levels of P24 expression in CLAT with Nef compared to CLAT without Nef. And please also note that similar results were observed with other LRAs and another independent CLAT clone. We are then further interested to understand what are the mechanisms underlying Nef latency reactivation. Could this be due to Nef ability to enhance TNF expression? In this regard, it has been previously described that Nef induces TNF-alpha gene expression, resulting into enhancement in TNF-alpha expression, which may enhance latency reactivation in our case. Alternatively, Nef reroutes ADAM17, which is a TNF-alpha converting enzyme, from the cell surface to intracellular compartments where it mediates intracellular cleavage of pro-TNF-alpha, resulting into enhancement of TNF-alpha expression, which again may enhance latency reactivation. And also, Nef enhances formation of extracellular vesicles known as exosomes, which also contain Nef and activated ADAM17, which may activate ADAM17 in a similar manner to virion-associated Nef. So we began by analyzing our first possibility, which is Nef ability to enhance TNF-alpha expression, so its ability to induce TNF-alpha gene expression. To do that, we treated CLAT with Nef, CLAT without Nef, and Pareto cell lines with prostatin, and we observed higher levels of TNF expression in CLAT with Nef compared to CLAT without Nef and parental cell line. These results are in part in line with Nef ability to induce TNF-alpha gene expression. To verify that Nef ability to promote TNF-alpha expression is associated with the enhancement in latency reactivation, we treated these cells with prostatin with anti-TNF-alpha antibody, and we observed that treating these cells with prostatin and prostatin with anti-TNF-alpha antibody is associated with marked decrease in P24 expressing cells compared to cells that are treated with prostatin alone. These results indicate that Nef ability to promote TNF-alpha expression is indeed associated with increased latency reactivation. To test our second possibility, which is Nef ability to activate ADAM17, we treated this cell with TAPI2, an established inhibitor of ADAM17, and we observed again a marked decrease in P24 expressing cells compared to cells that are treated with prostatin alone. And finally, to test our final potential mechanism, which is Nef ability to enhance exosomal formation, we treated these cells with prostatin and prostatin with GW4869, which is an established inhibitor for exosomal formation. Similarly, we observed marked decrease in P24 expressing cells when you treat these cells with this exosomal formation inhibitor. To validate this observation, we generated exosome-containing Nef by transfecting 293 T cells with plasmid-containing Nef, collect supernatant, isolate and quantify them through previously established methods. After that, we treated CLAT without Nef with the exosomes containing Nef and these two negative controls. We observed higher levels of TNF expression as well as P24 expression when these cells were treated with Nef-containing exosomes. To further validate this observation, we simultaneously stained for TNF expression and P24 expression, and we observed that cells that are expressing TNF are the ones also that are expressing P24. And so these results suggest that exosomal Nef also possess the ability to promote TNF expression and is associated with latency reactivation. And so Nef enhances LRI-induced viral reactivation from latency through a TNF-alpha mediated mechanism. Results are consistent with prior studies indicating that Nef can induce TNF-alpha expression and promote the formation of TNF-alpha containing exosome, which can stimulate latent infected cells in an autocrine or paracrine manner. Our work highlights a novel role for Nef in modulating latent HIV infection that may inform efforts to target viral reservoirs. Thank you.